Welcome to the Musella Foundation's Brain Tumor Webinar Series. It's my pleasure today to introduce Dr. David Needham and Kate Onk, co-founders of DNKO. Dr. Needham is a professor emeritus in the Department of Mechanical Engineering and Materials Science at Duke University and professor of translational therapeutics in the School of Pharmacy at the University of Nottingham. He has studied lipid bilayer membranes for more than 40 years, and this work led to his invention and development of Thermodox, which is the topic of our webinar today. Kate Onk has spent over a decade in technology startups across multiple industries, with a focus on building data infrastructure and analytics teams. Kate's personal mission to improve outcomes for brain cancer patients is driven by her personal experience caring for her father during his battle with glioblastoma. In addition to her role as co-founder and CEO of DNKO, Kate also serves on the U.S. board of Our Brain Bank. David and Kate, take it away.
Thank you so much for having us, Al and Vanessa. It's really wonderful to be here. And we really look forward to sharing our journey to this point and also our story about Dinko. So wonderful. I will kick it off. So we like to go by Dinko, which is our initials DNKO. We just pronounce it Dinko, which makes it a little bit more fun to say. And we're really focused on brain cancer right now. And we were established in 2023 to build upon a 25-year scientific and clinical history, which we'll start to talk through today. It's July 18th. We're very happy to be here with the Musella Foundation. Vanessa already gave us a wonderful intro, so I think she largely covered what we have here, which is fantastic. David and I both lost our parents to cancer, which really fuels the work that David has done over the past 47 years in research, as well as, kind of my pivot here over the last couple years. And we really look forward to kind of walking through our story today. A theme that is really at the forefront of what we do here is what we realized has been full circle. And we'll kind of explain a few of those kind of themes today and how it's circled up. But we also encourage you to kind of reach out to us if you find yourself hearing this presentation or learning more about our work and finding a full circle element coming to the forefront for you. Please reach out, as we found that really a resonant signal and an indicator that we're on the right path here with the work that we're doing. Wonderful. So how LinkedIn led to Dinko, it's funny. We mentioned my background in technology and working in analytics. And I was actually working at LinkedIn at the time when my dad was diagnosed with a brain tumor. And so I took family medical leave of absence, went to Cleveland, and was there for months on end. Coming out of that, obviously, I still use the LinkedIn platform. I joined the Our Brain Bank board, and I happened to be on LinkedIn one day in March of 2023 when I came across an article that said revolutionary brain tumor breakthrough unveils promising new drug treatments. And I thought, well, let me read this. This is exciting. So I read through the article, and it said Professor David Needham at Duke University invents this new formulation. And I thought, well, let me just see if he's on LinkedIn. And I sent him a connection request shortly after reading the article. And then David responded, I feel like it was within 10 minutes. And so we have the screenshot here of the message. And it's great to kind of look at this now over a year later and just see how kind of true we were to our mission and what we're trying to do to really benefit patients. And so I had mentioned that David and I met in March. And so over the first kind of 6 to 8 weeks of us kind of getting to know each other and also learning about his extensive history with cancer research and treatment development, at the time, the article had been focused on his niclosamide formulation. So that had been a large kind of focus of our conversation. But I had remembered hearing about Thermodox, his initial invention of the low temperature sensitive liposome doxorubicin. And one day was a Sunday evening here, as we can see. We're actually discussing like Dinko logo variations. That was the subject of this email thread. But I happened to be doing a top-down search, just thinking, okay, what are all the ways that they're treating brain cancer right now, to really make sure I'm aware of kind of the full extent of everything being offered to patients. And that's when I came across LITT, laser interstitial thermal therapy. And when I saw the, temperature ranges that it reaches, I was like, oh, wait a second. I remembered that Thermodox needed roughly 40 degrees Celsius, a couple degrees beyond that. But that really stuck out to me as something. And so I had emailed David right away and said, hey, this caught my eye. Could this be something we consider? Where things get really exciting is when we start diving into the literature. And one of the coolest things to find was the fact that the first in man study of LITT in 2013 was actually led by my father's surgeon that I met in 2019, Dr. Andrew Sloan. And so that was really exciting because I got to know Dr. Sloan very well. You know, my father was in the hospital for months at a time. And so it wasn't just that I met him, you know, before the surgery and after. There was, you know, weeks of continued engagement there. And so he was somebody that I really got to know out of this experience. And so to see that he actually led the work there was very exciting because it meant we are now uniting the inventor of the drug with the key surgeon that led to FDA approval of LITT. And this was all via personal connection that I could bring to the table. And the most exciting part about this is that once we get through our preclinical studies, Dr. Sloan is willing and very excited to lead the phase 1 clinical trial at Piedmont, Atlanta. Pass it over to David here.
Yes, so I just spent 36 years at Duke as a professor and retired at least off the Duke payroll on the 30th of June, 2023. And we started Dinko on the 1st of July. So having finished 36 years at Duke, I plan to spend the next 36 years at Dinko. So our vision and mission, we came up with these short sentences. Basically, our Dinko vision is that brain cancer patients will have access to a clinically more effective treatment. And this is kind of our ethos, that it prioritizes patients before profits. And then when tested, it may even be curative for the right sized and lit heatable tumors.
This means that our mission is to combine existing and proven technology, David's invention of the low temperature sensitive liposome doxorubicin, which you'll see referred to as LTSL dox as well, and laser interstitial thermal therapy, LITT for short, within the brain cancer community. And also to unite like-minded clinicians, as we've seen with Dr. Sloan, to test it, evaluate it, and if it shows promise, bring it to their patients as soon as possible. We'll share later, but we do have a little song focused around Dinko that if anybody wants to listen to, we'll be sure to get out there.
So just to introduce what the technology is, LTSL dox plus hypothermia addresses basically any cancer that can be heated. And in the figure there from the paper by Petra Koch and Johannes Creasy, there are 15 different tumors that could be heated, and therefore there are 15 different tumors that could actually be treated. In the blue boxes, we have the primary targets that we're focused on now is brain cancer, but we also have collaborators looking at the bladder and the prostate. And then in the pink boxes are the ones that were already tested by us at Duke and Celsian. Liver was the primary liver, and then sarcomas were in the extremities, and chest wall, which is a cancer that occurs after mastectomy of breast cancer. And then over on the right is a schematic that we had made up in 2000, so 24 years ago now. And what it's showing is that the tumor is warm to 41.5 degrees. And as the thermal sensitive liposomes come through the vasculature that leads into the tumor, as soon as they hit that temperature, they immediately release their drug, which is then permeates the whole tumor. We have actually videos on the website that show a tumor filling with doxorubicin, which is fluorescent, within 20 minutes. And so I've been known to say in presentations, the only way to deliver drug throughout the entire tumor is to release it in the blood vessels of the tumor.
Wonderful. So it's a real honor here to share some of our additional collaborators and our advisors here that really shepherd us on this journey. And so we have our 3 preclinical and clinical collaborators that are implementing and advising on this technology. They have each been at the forefront and are early adopters of one or both aspects of this drug device technology for decades. They aid us in developing our optimized, more effective clinical protocols and will be the first clinicians that provide service in these clinical trials. So that's Dr. Sloan, who I already mentioned, who is the Chief of Neuroscience at Piedmont Atlanta, Dieter Hammerich, who's a professor of pediatrics at MUSC. He's really carried the torch for a lot of preclinical studies with Thermodox over the years, including some canine studies with Thermodox in the brain already. And then we have Jack Hoops, who will be leading our preclinical pig study that'll be happening at Dartmouth, which I'll speak to in a moment.
Yeah, and our 3 industrial collaborators that guide us in intellectual property and legal, as well as regulatory strategy with the FDA and manufacturing the LTSL docs. It's really a pleasure to introduce Mike Tarduno, who led the whole development of Thermodox from somewhere around 2004, 2005. So he had 17 years as the CEO of Celsion. He's now the chairman of the board, but they've come back, and again in this full circle issue that we're seeing here, he's now been very helpful and started to work with us, even though he's the chairman of the board at Immunon. He's still very keen on seeing this technology through. And Nick Boris, MD, is the 16-year veteran of the medical studies, the clinical studies that were carried out. He's now president of NB&C Consulting. And so we're working closely with Nick too. And another full circle was Jeff Mills. So Jeff is our patent attorney. He's a partner in a DC law firm, Medler, Farrow, Woodhouse, and Mills. And it turns out that Jeff was my PhD student back in 1997 to 2002, and he worked on the LTSL docs project for his thesis.
Thank you, David. Wonderful. So as I had just kind of mentioned, the Musella Foundation awarded a $50,000 grant, which will support our swine study at Dartmouth. And this will be starting in August of 2024. This was very exciting for us because it was also announced on our 1-year anniversary of launching Dinko just a couple weeks ago. So that was also exciting. And this money will be used to really understand some of the thermal dynamics that we're looking to in this pig study, which will inform if we either move forward with a canine study next, or if we go directly into a phase 1 study in human recurrent glioblastoma patients. And our collaborators are Dr. Hoops and Dr. Evans, as well as Dr. Tavakapi. And to ensure a pathway to a phase 1 clinical trial, we've estimated that we need to raise roughly 2.27 million. This will enable us to continue any preclinical studies that we need to do after the pig study, as well as manufacturing a new supply and bringing 12 patients into a phase 1 study. This would be at Piedmont, Atlanta, led by Dr. Andrew Sloan, who, as I mentioned, was my dad's surgeon back in 2019. And so it really, it's very rewarding to be at this point where we can kind of bring something to patients. And as we mentioned, by working with the Musella Foundation, we're able to receive funding for this work via tax-deductible donation to the Musella Foundation under the Dinko Research Fund, which they have set up to help us raise this 2.27 million. It's really just an honor to work with them because they have such extensive experience, personally battling this from the patient caregiver perspective, but also the decades of understanding Al has kind of to the regulatory space, as well as the medical advisory board that has reviewed our protocols and can offer an extra set of eyes on some of our clinical ideas, which is just really welcome in this environment. And one other thing we just want to mention here about donations is I know a lot of companies offer donation matches, or they have a matching gift program where if an employee donates to a 501 501(c) or C3 nonprofit, they can receive a matching donation from their employer. And so we do encourage you to see if that's something, because it's a great way to amplify your contribution to research and to benefit brain cancer patients. So thank you. We appreciate the time here today and the platform to really explain our work and some of the kind of key moments that we think led to the momentum here today.
Thank you so much for that presentation and sharing all of that background information. You really have come such a long way in a short amount of time, and we're so excited to see what's to come. I just have two quick questions. My first, for all the dog lovers out there, you did reference a canine study. So can you just give a little bit of information about that?
Yeah, so that is kind of pending what we see from the pig study here, and to understand, does it make sense for us to go into canines first that have spontaneous growing brain tumors, which we could then understand, some of the other dynamics that we're not going to get out of the pig study because these will just be healthy pig brains. So there is kind of one angle there. On the other end, we do want to give it to patients. So, we're going to see what the data tells us and kind of go from there. There could be a pathway where we pursue both, right? We don't need to let a canine study potentially keep us from going to a phase 1. We could proceed with both things if we see enough kind of positive data coming out of the pig study.
Okay, so the canine study is going to be actual canine patients.
It would.
We're not injecting dogs with
no.
glioblastoma in order to test this treatment. Okay.
And actually, now that we're collaborating with Jack Hoops, he's a bona fide DVM radiation oncologist, and he gets canine patients up at Dartmouth. And they're not just brain cancer dogs. They have many other cancers like, facial or head and neck cancers. And he said to us a few weeks ago in a conversation that he wished he'd had Thermodox available to him when he was actually doing radiation treatment on one of the dogs. And so we've now got a procedure in that we can get him some of the vials that are available from Celsion that were returned from the clinical trials to be used for the animal patients. So we're seeing this, like I said, there are 15 places that the tumors can be heated, and he's actually a real expert on hypothermia as well as radiation oncology. And so he's got the heating devices to warm head and neck cancers and sarcomas, as well as brain cancers, in collaborating with Linton Evans. And so what we're expecting is that once he gets this batch of vials of Thermodox, he'll start doing actual clinical treatments with owner's consent, and then that could develop into a proper canine trial, and then it could also go veterinary.
Great. Yeah, thank you for that information and context. And you mentioned previous research. So Thermodox has been used in humans previously, used safely.
Yeah, the Celsion, they licensed it in 1999. And then immediately started they immediately started a prostate cancer trial using a benign prostate hyperplasia heating device. And that went through a phase 1 trial, and that's where they established the maximally tolerated dose and the safe dose as 50 milligrams per meter squared. They went up to 65, came back to 60, and then decided, or 55, and then came back to 50 as the MTD that they wanted to use as a clinical dose. And so that was probably 20 patients initially in 2003 to 2005. And then it moved into another 20 or 30 patients in chest wall recurrence at Duke, phase 1 and phase 2 trial. And at the same time, Celsion decided to move over into primary liver cancer and do a phase 1 trial in that, which actually showed 150% progression-free survival in a range of tumors in that phase 1. And that's why they were so excited about it. And then they set up a full-blown phase 3 trial in liver cancer in 701 patients in 14 different countries in 71 different sites. And so half of those got Thermodox and half of them got the regular radio frequency ablation. And then they carried on and did a second phase 3 study in an optimized protocol. And so in talking with Mike, he was saying that over 800 patients have had Thermodox with various primary liver, chest wall, prostate, but also metastatic bone cancers. They set up quite a few small trials with various collaborators, and in that time, from 2003 to 2021, it's also reported. The HEAT study is reported on clinicaltrials.gov, and there are very few adverse events that were serious. Doxorubicin itself can be toxic, obviously, if it's infused. But the whole point of putting in a liposome was it actually reduced its toxicity. And so there is a lifetime dose, but that's something like 300 to 500 milligrams per meter squared. So in these initial studies, we're expecting it to be safe, and have the Celsian has the data, showing that.
Okay, and why doxorubicin as opposed to, another drug candidate? Theoretically, you could use the same liposomal.
Sure, no,
delivery.
thought of that, and other people have published on cisplatin and different things. But remember, this is not today. This is 1990. And liposomes were only invented in the late 70s or early 70s to late 70s, 80s when the stealth liposome came in, 1987, 1988. And so when we were looking for a drug to put in a liposome, we basically took the existing liposome, and my invention was to make the membrane permeable as a function of temperature, actually 41.5 degrees, which is just above body temperature. So we piggybacked what we did on what was available. And so it's kind of interesting that even today, I'm not sure I would switch the drug, because when you look at what this particular drug does, chemically, it has a property that it will go through lipid membranes. And if people want to go to our website and look at that video that was made in Dr. Dewhurst's lab, you'll see the whole tumor filled with drug within 20 minutes. The drug's fluorescent red, and the whole tumor turns red. All the drug is in the nucleus of every cell, and the reason it can get there is because it is so membrane soluble and passes through the tumor. So, sure, if there are other drugs that we could consider, but then we'd have to start again, and we're already 25 years in.
Okay.
The real beauty here, I think, of picking up this technology again at this point in time, and one thing I realized we didn't really mention at all in the slide deck, is the advancements in real-time thermometry, which LITT offers. And so we didn't dive too much into LITT here, or kind of the technology behind the different heating applications that have been used over the 25-year history of Thermodox, but that becomes kind of the key thing that, we evaluate as what was missing in the past, was that real-time thermometry feedback loop to say, hey, we set it to this temperature, we know this tumor pixel point has been at this temperature for X amount of time, thereby we think that this amount of drug should have been delivered, right? You can really start to kind of do that analysis once you know that the heat is actually being delivered, and you have that feedback loop to know that it's being maintained. And so that's why we're so excited. And maybe that's like another topic for us to kind of dive down in the future, is some of the real mechanics that we're looking to pull together here in this study.
Yeah, and
Yeah,
actually,
the community, the hypothermia community it's at least a 50-year-old community, right? They were doing it back in the 80s. Dr. Dewhurst was in Tucson with other people. In fact, Jack Coops was with him. There's another full circle. And so that technology has been developing and advancing. And then, like Kate said, with the ability to actually measure the temperature, it's like measuring the dose. Whether it's a chemotherapy or whatever, you want to measure the radiation dose or the heat dose. They can measure the heat dose now to very precise locations within the MR imaging. And so you may say that in 1996, Thermodox was ahead of its time. But now its time has come, and we've got some really top experts that have been doing this for a very long time, and they're really excited about taking it forward.
So based on those, I think you mentioned 2 phase 3 trials, did they ever seek FDA approval?
The first one, the heat study, got within that distance of actually being approved. What apparently happened there was there were multiple sites. Like we said, we're only working in one site for brain cancer. We want to be able to control this and work with the clinicians directly. But they had 71 different sites and used different heating devices. And the radiofrequency ablation ablates tumors. It will cure 3-centimeter tumors. But the decision was made to go for the 3 to 5 to 3 to 7 centimeter tumors that were never really treated, to see if Thermodox could do something there. But it turned out from actually recent calculations that I've done that the edge of those tumors that actually are the place where the cancer it develops again were never really heated successfully. It was a good idea to try it, and there was a big market for it. And if it had been successful, it would have been pretty amazing, but the physics was just not there. And so that just missed getting approved. There was actually a subgroup in Korea, South Korea, that did get progression-free survival that would have got it approved. 245 patients actually did see that level, but then the other 200 or 300 brought the average down. And so the average was just below what they needed. And then in the Optima trial, where they heated for 45 minutes, again, using the radiofrequency ablation, did not necessarily get out to the edge, although they were seeing some responses. And that trial ended in 2021 without being fully resolved.
It's just so interesting. You know, I've heard neuro-oncologists, even on our Musella Foundation webinars, you know, a lot of them say it's their great fear and worry that we've had great drugs over the years that have failed in trials because either we weren't using them the right way or we weren't using them on the right patients. And so it is really wonderful to see what you're doing and looking back and thinking about an old therapy in a new way, especially with the new technology we have nowadays. So I'm really excited for this, just on a kind of a time frame. You know, obviously there's a lot still to be done, but do you think this will reach human trials anywhere in the near future?
It's definitely our hope. And this pig study is what will really inform that. You know, if we do need to do a canine portion beforehand, we do know canine glioma patient recruitment can be slow. And so, you know, that's what kind of could change this window. But if we see kind of positive signals at the pig study, it's really then just a matter of the funding to be able to push it through. I think, you know, as far as having our collaborator, Dr. Sloan, ready to lead it and the site chosen, you know, we have all of those pieces in play, then it just becomes down to seeking funding essentially.
Great. And that's the perfect place to end today's webinar. I know you shared the location on the Musella Foundation website where people can make donations toward this really important work. And just to reiterate, the website address for that is virtualtrials.org/donate. Thanks so much.